Attendees are in listen-only mode.
All right, good afternoon, everyone, and welcome to our inaugural HDSA Research Webinar Series. My name is George Yorling. I'm the Director of Medical and Scientific Affairs at HDSA in our national office here in New York. And I just wanted to welcome you all for participating. In an effort to increase our research communication efforts, we're really excited to bring this new forum to you as an opportunity to have a regular interaction with the scientists that are actually participating in the HD research around the world. So just just move forward. Some ground rules for today. This is much like the Caregivers Corner webinars that hopefully some of you have attended in the past. There will be an opportunity at the end to ask our presenter questions, and they will be answered by him. You can send your question at any time during the presentation. Just go to the control panel that appears at the right hand of your screen and type in your question and click send at any time. And as we said, at the end, we will address as many questions as we can. It's important to note that all of these webinars today and all the future ones will be recorded and archived on our HDSA website. Within 1 week, you'll be able to find this webinar and view it or pass along the link to others that you may think might find this interesting. To access this presentation, just go to www.hdsa.org/researchwebinar to view the recording. So with that, today I'm really excited to welcome our guest, Dr. Jamshid Arjaman. Dr. Arjaman is currently the Director of Basic Research at CHCI Foundation in the Princeton, New Jersey office. He's been there since 2005 and is one of the things areas of research that he's become an expert in the field is stem cells and induced pluripotent stem cells, or iPSCs, all of which you'll hear Dr. Arjaman present an overview of. Dr. Arjaman received his bachelor's degree from UC Berkeley and in molecular biology and psychology, and followed up with a PhD in neuroscience from the University of California at Los Angeles. So without further ado, I'll turn it over to Dr. Arjaman. The title of his talk today is Stem Cells in HD Research and Therapeutics.
George, thank you very much for this very kind introduction. It's a really great pleasure to be a speaker at this seminar, and I'm really thrilled to be the first speaker of the series, especially having seen the potential speakers you're going to have down the line. Before they set the bar too high, I'll be able to give my presentation. Hopefully it will be of interest to the audience. So to get started, what I thought I would do today is kind of give an overview of stem cells. There are different types of stem cells, and I thought it'd be interesting to review the sources and what they are. They're always in the news, and there's always some of them are controversial, or there's some ethical questions. Hopefully we'll be able to address that in the first part of the talk. I will then discuss some of the potential therapeutics applications of stem cells, and this is still very much a field that it's in its infancy. However, there's stem cells do hold a lot of promise, and so I kind of wanted to highlight some of the studies that are ongoing. And finally, I'm going to talk about the use of stem cells in research and development. And as Director of Basic Research, this is really the area that I focus in. And I'm very excited about the ability to use human HD stem cells to understand the disease better, and I'll try to showcase one of the larger programs that we're helping coordinate with NINDS. You'll probably recognize a lot of the investigators. They're former HDSA coalition members, so you'll see these names that you're familiar with pop up again, and they're doing some really incredibly stimulating and interesting work that I'll try to describe. So before I get too far along, I know that you kind of are giving me a hard stop in 45 minutes, so I thought I'd start off with the most important slides. This is the acknowledgement slide. There are a lot of people that have been involved in doing HD-related stem cell research. I'm noticing that the slide is getting incredibly crowded, so I'm going to probably have to make different acknowledgement slides depending on where, what kind of research I'm trying to highlight. But here it's still trying to include everyone, a lot of groups, companies, as well as I need to thank, my collaborators and colleagues at CHDI, a lot of advisors and experts in the field from other areas outside of HD that have given a lot of input into the design of some of our programs, and most importantly, the HD families and patients who participate and who are always the inspiration for the work that we try to do. So, let's start off with what are stem cells. So the definition of stem cells, hopefully you can see my pointer here, I'll be using this a lot. They're unspecialized cells, and they're capable of growth and continuous growth in the right proper medium. And they also have the ability to turn into just about any organ. So this is essentially the definition of a stem cell. And so what I have here is a cell in a dish. It can grow and you can continuously regenerate so indefinitely. So and then it can differentiate into all the germ layers or all of the different tissue types. And these type of stem cells are called pluripotent. There are more restricted stem cells, and we'll go over those a little bit later. But if you're very interested in learning about the basics of stem cells, the NIH has a very good website that I've highlighted down here that you can go visit, and it explains some of the basics. You can always review the source of stem cells and what they are. So, and one of the points about that every stem cell can differentiate into any tissue type is actually one of the hallmarks of how people test for stem cellness. So if they generate a stem cell, quite often what they will do is they'll implant some of them in a rodent model, and that actually forms a tumor, or it's called a teratoma. And if you were to dissect it and cut through it, you will see all of the different layers, all the different tissues represented. There are teratomas that form teeth and hair and muscle and heart, and pathologists can actually identify this. And so here is kind of an example of muscle and fat tissue and even neural tissue is kind of engrossed in this teratoma, in this tumor. So there are other types of stem cells. So there are just about every organ in the tissue has resident stem cells that can help it regenerate. It used to be thought that the brain, once you develop the number of neurons in your brain, that's it, it's static, you can't regenerate new neurons. Well, it turns out that there are pools of neural stem cells that live in the brain. And you may have heard about effects of exercise or even some antidepressants that stimulate the growth of stem cells in the hippocampus. There's another big area of growth, and this is in the ventricular zone, the subventricular zone area. And these neurons generally, at least in rodents, they migrate and they repopulate the olfactory bulb. This is basically the nerves that innervate your nose and your sense of smell. And obviously for rodents, this is a very big sensory organ. So there's you can really study this well in rodents. Just to recap, the neural stem cells are limited. They're multipotent, so they can't give rise to every type of organ. They're restricted and they can only generate the neurons and then the accessory cells that can be found in the brain. These are the oligodendrocytes that form the insulation to the nerves, as well as the astrocytes, which are kind of maintenance cells that clean up and a lot of the debris and that provide nutrients in support of the neurons. There's another big source of stem cells, and you may have heard about these. These are called mesenchymal stem cells, and these are generally isolated from bone marrow, and they also have limited capacity in their differentiation. Now, the interesting thing about these mesenchymal stem cells is that normally when their therapeutic application, and I'll cover that a little bit later, is that they may be able to tone down the immune system a little bit, and so and they may also secrete factors that might be protective to tissues and organs. So the mesenchymal stem cells are often also called MSCs, and the literature actually, there's a lot of different studies that refer to MSCs. There are things called multipotent cells they're actually multipotent stromal cells. This is the same acronyms. So those can be derived from tissues like fat, from liposuction. So these are a little bit less invasive sources of these MSCs. And then you have the embryonic stem cells. And so these are derived from embryos. And so you start off with an egg and a sperm, and once the egg is fertilized, it goes through several rounds of cell division. And what happens is after about 8 to 10 days, you have this blastocyst, and in the blastocyst, you have a bunch of cells called the inner cell mass. And these inner cell mass can then be isolated, and under the proper culture conditions, with the right factors, they can actually give rise to these stem cells. These are called embryonic stem cells. They are pluripotent. Under the right conditions, they can divide indefinitely. And because during the process of their generation, the embryo is destroyed, that's cause of some concerns and some debate as to whether there should be used in research or not. And as you might remember, during the Bush administration, President Bush banned the use of federal funds to generate any new embryonic stem cells. There are some laws, strict laws that kind of limit the generation of human embryonic stem cells. You cannot create an embryo for the sole purposes of making human embryonic stem cells. However, when Obama became president, he signed an executive order, again allowing federal funds to be used to do research with embryonic stem cells. There's still some strict guidelines on how they can be derived, and the web link I have here actually outlines some of the ethical guidelines for the generation and use of human ES cells. Now, how could ES cells be generated? So I just wanted to review here. Most of the audience is probably familiar with the cause of Huntington's disease. Huntington's disease is caused by a mutation in the Huntington gene. There's a CAG expansion and over a certain threshold that causes the disease. So because the gene has been identified and it can be tested for, there is a process called pre-implantation genetic diagnostics that allows embryos to be formed in vitro and tested, and the families who undergo PGD IVF can then be made sure that their children will not carry the mutation. And the process is that the eggs are harvested just like any sort of in vitro fertilization from the mother. They are fertilized in vitro in a dish, and then once they are fertilized, a couple of the cells are isolated and they are tested to see if they carry a mutation or not. And the embryos that do not carry the mutation can then be safely transplanted in the mother for the family's reproductive needs. Now, obviously, the embryos that carry the mutation are not going to be implanted, and so generally these have been destroyed, or but with the proper consent and depending on the laws of each country, these these embryos can be used to make disease ES cells, human embryonic ES cells. So there's several HD cell lines ES cells have been made, but I just want to highlight a catalog. This is a company in Australia. They were formerly known as Sydney IVF. They're a very large in vitro fertilization clinic. They have been rebranded as Genea, and they do a lot of this PGD IVF procedure for a lot of different genetic disorders. And here what I want to highlight in their catalog is they have generated HD ES cells for research purposes. And what you can see is their process is rigorous, and they're informed consents are done in such a way that they are in line with the current NIH guidelines. So they've been submitting the cells as they produce them for review by NIH. And so the ones that are highlighted here have already gone through the approval process. I believe these 2 cell lines are in process of being reviewed by NIH, and I'm certain that they will be approved. But one of the things for researchers, if anyone are listening, these cells are available from GenAI, and they can be obtained for research purposes. Now, there's a caveat here. Every country has also limitations on what they allow, what kind of research they allow. So for the time being in the U.S., federal funds can be used to do research using properly consented and NIH-approved stem cells. In Germany, for instance, there is a ban on the use of any stem cells that were derived after 2007. So German researchers can't use these lines from GenAI because they were derived starting 2008. So down below, I've also highlighted the website for the different stem cells that are available through GenAI. So next, I want to talk about this fairly recent discovery. These are called induced pluripotent stem cells, and this discovery there was a paper published in 2006 by Shinya Yamanaka's group, and I have his picture here. He won the Nobel Prize for this work just last year. And really, this discovery really revolutionized the whole stem cell field, and it's really remarkable. What they did was they were able to take skin cells, and these are from adult animals, and reprogram them into essentially these stem cells, embryonic stem cells that are pluripotent. This is a really remarkable advance, because what it allows and soon after they published this paper, which was done using mouse cells, there was a paper that they published showing that this could be done using human cells, and Dr. Jamie Thompson in Wisconsin also published a paper about the same time showing that this could be done in human cells. It's been replicated by a lot of different groups. There's a lot of different methods now for doing this kind of reprogramming. But essentially, what this allows researchers to do now is to do a very small skin biopsy. You can actually reprogram blood cells as well, and you can convert them into these stem cells. And then you can differentiate them into different organs to study disease. You can use them to do drug screenings. This is a really powerful tool. You can now use bona fide human cells for research purposes. So how can these really impact research in HD? Well, first of all, the iPS cells don't carry with them the baggage of the ethics and the legality. You absolutely need to properly consent the donors, the participants, and make them aware that the blood they're giving or the skin graft, the skin biopsy they're giving are going to be used for iPS derivation. So there's definitely very well laid out guidelines on how to do this, but the ethics of whether an embryo is being destroyed is no longer applicable here. These cells are derived from skin cells or blood cells. Because it's relatively easy, I'm now kind of saying it's easy. It's easy for experts to reprogram these cells, but because it's a technique that can be done in the laboratory fairly easily, it's now possible to actually generate iPS cells from from very large populations or large groups. And just to kind of highlight this, I have a very small screenshot down below here. You won't be able to see it too well, but the NINDS has generated a repository at Coriell, and this is they deposit. There's about a dozen or so iPS cells that have been deposited in Coriell, and about 60 more cell lines that are probably in queue of being quality controlled and deposited. And these cells are going to be available for the entire research community to use. And just as a highlight, I visited the New York Stem Cell Foundation recently, and they've almost automated this entire process for derivation of iPS cells. So, the lofty goal of these cells is that at some point they will have some therapeutic application. And because the cells can be derived from the patient, the ideal goal is that at some point we're going to be able to have personalized medicine. We're going to be able to take the cell from the individual, do whatever correction needs to be done if necessary, grow a new organ, and then transplant it back. This is still years to come, but this is kind of the goal of where this field is headed. And using this, I'm going to segue into the second part of the talk, which is the potential therapeutic application. And I just wanted to highlight this concept of using iPS cells to treat a disease. This paper was published a little over a year after Dr. Yamanaka published his seminal paper for showing that iPS cells can be made. So this was published in December of 2007. And what Yanish's group did was they took a model of sickle cell anemia, which is also a genetic disorder, and what they did is they generated iPS cells from the mouse, and they were able to reprogram these, and then they did a gene correction of these iPS cells in the dish. They then grew up these cells into larger quantities, and they did a partial differentiation. Now, sickle cell anemia, for those of you who might not be familiar, is a disorder of the blood cells where they kind of take on this sickle cell shape and they clog up the small arteries. And the blood cells, of course, are generated in the bone marrow. So what you can do then is you take the mouse that has sickle cell anemia and you can irradiate it to kind of kill the mouse's endogenous bone marrow, and you can then transplant these corrected cells into the bone marrow. And essentially what you end up doing is you cure the mouse. This is a remarkable proof of concept study. Here you see the arrowheads showing these sickle cell shaped cells, and they are not found in the treated cells. So again, what's really kind of the goal of any of this research is that you're going to be able to take donor cells, make iPS cells, do any type of gene therapy that's necessary, grow the organs, and then put it back in the donor. But the word of caution is that these studies were done in mice. The process really has a long way to go before it can be adapted for any sort of a human trial. So sickle cell anemia is not being treated this way clinically, even though this paper was published in 2007. And so one word of caution might be that just because somebody publishes a really exciting paper where they've done some treatment through a mouse doesn't mean that it's going to be immediately applicable to clinical trials and human application. So because of that, because there's been such a hype about the use of stem cells in therapy, there's been a lot of, kind of bogus companies that have popped up offering these miracle treatments for a variety of diseases. And they're really preying on populations that are desperate for treatments. And so the International Society for Stem Cell Research, the ISSCR, kind of recognized that, and in 2008 they published the guidelines for clinical translation of stem cells. And they've also generated this really great website. You can find that www.closerlookatstemcells.org, where it kind of highlights the type of questions that anybody should ask when they're evaluating any sort of treatment. And this is really important that people kind of review this, and they speak to physicians to kind of see if stem cell treatment is really an option for whatever their affliction is. But the incentive here, and this is kind of the guidelines were generated to basically restrict and make people aware that there are some companies, offshore companies, that are offering treatment that really are not treatment. They can possibly even cause a lot of harm. So there's, stem cells. I described several different types of stem cells, and these can be potentially used in different strategies for therapeutics. So I took this image from a review by Dr. Goldman's lab, and I just added one more possible avenue here. I'm just going to kind of quickly review these. We mentioned that the mesenchymal stem cells are they don't have the ability to, they're multipotent, so they cannot become nerve cells. There's some debate about this, but in general, the idea of using the mesenchymal stem cells for treatment is that the mesenchymal stem cells are either going to act indirectly by quieting down the immune system, or they may be releasing some factors that are going to help sick neurons live. And of course, these cells can also be engineered to secrete factors that we might think are critical for the survival of neurons. So that's one approach, and these are the MSCs. Another approach might be to use gene therapy to kind of like stimulate the adult neuronal stem cells that are already resident in the brain. So if you can actually generate more neurons and you can guide them to go to the appropriate place and you're basically harnessing the brain's own source of stem cells. Now, one thing to consider here is that in the case of HD, these neurons will also carry the mutation. However, because they are younger and newer, maybe they'll last longer and they might be beneficial. So I'll highlight some studies that have been done in this area. And then there's also a fetal brain transplant. These are not necessarily stem cell therapeutics, but it's close. These are the actual physical injection of fetal brain into the brain as a replacement therapy. And finally, there's the use of the possible use of embryonic stem cell or the iPS cells, where the cells would be grown and differentiated into the relevant neurons or the different type of cells we're trying to replace and injecting those. So just here, I'm going to highlight a couple of challenges that are facing any type of kind of transplantation therapy or stem cell therapeutics. So these are the what, when, and where kind of questions. What kind of cells should be transplanted? So as I mentioned, the neuronal stem cells can give rise to neurons, oligodendrocytes, or astrocytes. These cells, all of them in HD carry the mutation, and we don't know if the disease is caused because of all of them are sick and if just replacing one type is enough or not. If we also, another word of caution is if the cells are not properly differentiated, if they are too stem cell-like, as I mentioned, they can form tumors, and this is an example of cells that were injected in a rat brain when they were not fully differentiated, so they continue to grow and they form this giant tumor that's highlighted in darker purple. When should the cells be transplanted? That's another big question. So there are some guidelines for the fetal transplants that have been that clinicians have come up with, but with regards to the graft, is when is the best time to do the implants? So if you are trying to give mesenchymal stem cells that are producing growth factors to prevent the death of neurons, the idea would be you might want to give them early on. If you are trying to give a graft to replace cells that are gone, there might be a reason to want to do the injections a little bit later. But this is something that's really needs, we don't know exactly when this should be done. And also, where should the cells be transplanted? So there's been a lot of focus on the striatum or the basal ganglia. This is the area that degenerates the most. However, there's degeneration everywhere. And Huntington is expressed in all the different types of neurons and the accessory cells. So is it enough if the injections or the transplants are made in just this area? Will it have a beneficial effect when there is disease progression throughout the brain? And so this is what the point of a lot of this research that's been going, and a picture is really worth 1,000 words. Most of the research is being done in rodent brains, and this is kind of the size of a mouse brain relative to a human brain. When you do injections on a mouse brain, you're coming in with a needle and you inject a bunch of cells. The equivalent, if you're trying to do this in a human brain, proportionally would be like putting a pipe in the human brain, which can't be done. So the migration that you see in a mouse brain can look very promising to actually be able to predict from the mouse studies about how this is going to work in a human brain is a very different story. The other kind of thing is that the cells, any cells that are transplanted, if we're trying to replace cells, they not only need to survive, and they need to make the proper connections. These cells are part of a very complex circuitry. They receive inputs from the cortex, from different brain regions, and they project to a bunch of different regions. So if we want to try to restore function, they need to connect properly. And so we don't know if this is actually going to be possible or not, but there's some really good research being done by a lot of different researchers using animal models, using a bunch of different HD mouse models, a lot of different techniques, a lot of different cell types, and all of these studies are going to help the clinical researchers kind of prioritize and better and decide what avenues are the most promising to take forward. So here I just wanted to highlight just a couple of studies. This was published by Dr. Goldman's lab, and it's about stimulating the endogenous stem cells. So what they did is they injected viruses actually in this area. These are called the ventricles. These are the fluid-filled cavities in the brain. And the viruses kind of infect the lining of the cell that kind of surrounds the ventricles. And they start producing these growth factors that promote the endogenous or the resident neural stem cells to grow. And what they've done is in an R62 model, this is a pretty aggressive mouse model of Huntington's disease. They showed that there's some marginal motor improvements in the mice that are treated with these viruses, and that it extended the lifespan by about 10 to 15%. Now, again, these are mouse studies, and just because somebody can treat and show a beneficial effect in a mouse model doesn't mean that this can be used in the clinic. So going to some of the mesenchymal type of studies, so here was a study where the human mesenchymal stem cells were injected in I believe it may have been a rat, a mouse model of Huntington's disease. And here what you can see is that and this is taken from their conclusion, the implants actually disappeared over the course of 2 weeks, and you can see the amount of green fluorescence. The cells were actually stained with this green marker, and you can actually see that the number of green cells disappears over time. So this is kind of an example of of graft rejection. So the animal's immune system is actually identifying the implanted cells as foreign and destroying them. So their conclusion was that we need to kind of really work out the what, when, and where of the studies. So then I just wanted to highlight the study. So these are actually MSCs that were derived from fat tissue. And again, other researchers are using different sources of the mesenchymal stem cells or the multipotent stromal cells to see if they have any beneficial effects. And finally, I wanted to highlight, this is where the MSCs have actually been engineered to produce some of these factors that we think are going to be beneficial. And what you can see here is the black line is the untreated mouse model, and the blue line is the mouse that has received these cells, and they do just a little bit better than the untreated mouse. Now, there's a lot of reasons why that might be the case. You might have rejection of the graft. The combination of factors that have been selected may not be the right factors. The mouse model may be resistant to any sort of treatment. There's a lot of things, but this is very promising that people are actually doing these kind of experiments, trying to figure out what is the best combination, what is the best timing, what is the best source of material to do this kind of research. Then I want to kind of like very quickly touch on some of the fetal tissue transplantation. These have been ongoing for quite a while, probably about 20 years or so. And they involve the transplanting of fetal tissue into the brain. And most of these studies have actually been done as very small open-label trials, so only a few, one or two possible patients in every study. The techniques are all a little bit different, so it's very difficult to make comparisons across the groups. Most of these studies don't have a proper control group because you can't ethically do a surgery and not put anything in a patient. And now there's some other difficulties with these type of studies is the scarcity of the tissue. So what you can see here is a cross-section of the brain, of a fetal brain, and there's these lumps here are called, the area that gives rise to the striatum of interest is actually called the LGE. And so what the newer studies are trying to do is dissect this area from the fetal brain and inject that into the patients. Now, to actually do this successfully, you probably need 2 to 4 donors for each patient. The tissues can't be stored for any length of time, so it's actually technically very difficult to do these kind of studies. Then there's other things that you know, are difficult to do in humans, is like monitoring how the graft is done. In a mouse, you can actually do the analysis. You can you know, cut the brain and actually see over time how is the graft doing. You can't do this with patients, so you have to use advanced technical methods, imaging methods that don't really tell you whether the grafts are integrated properly or not. But basically the overall you know, summary from all of these studies, and this is taken from a review by some of the experts in the field, Anne Roser and Catherine Bouchon-Lavie, is that there is some limited but transient improvement in some of the tests following these kind of transplantation. However, the disease continues to progress, so the transplants don't halt the progression of the disease. And more importantly, there was no adverse effects. So although in a couple cases there were some overgrowth of the grafts, so some tumors were formed, there were no deaths resulting from the grafts. So this basically, these kind of initial studies kind of prompted a larger scale clinical study that's been ongoing and is supposed to conclude, I believe, in middle of this year of 2013. This is called MIG-HD, or multicentric intracellular grafting in Huntington's disease. What they are doing is, this is a study carried out in France. Their goal is to do the fetal transplants in about 60 patients, and then evaluate the patients after about 20 months over motor tests and a bunch of other clinical tests. So if you are interested, you know, go to the clinicaltrials.gov for the results later on this year. Finally, I'm going to very quickly go over the uses of stem cells in research and development. So at CHDI, we're very interested in using stem cells mostly to model the disease. So we could learn more about Huntington's pathology using a human cell program, human cells, and also to use them to screen for drugs or to test different therapeutic strategies. And then also CHDI is very interested in contributing to the research of researchers around the world. So we wanted to kind of create a library and make any of the stem cells available to the community at large. So I'm just going to highlight one of the studies that's been that I'm involved with, and this is the HD-IPS consortium. It was initially funded by NIH in 2010 using ERA funding, and it involves a whole group of U.S. As well as European researchers, and their recent findings were published in 2012, and this is the heading of the paper. And just very interesting, their interaction is very different. What I showed you previously with the mesenchymal stem cells is labs run their studies using whatever type of stem cells, using different mouse models, and you kind of generate this body of literature using a bunch of different models. What the consortium is doing here is they agreed that they're all going to use the same cells at the beginning. So it's going to be easier to do these comparisons from lab to lab because they're all using the same starting material. So, and then each lab has expertise in different domains of Huntington's disease research. So they use those skills to kind of see if there are any differences in the HD cells versus the control cells. And if they find any differences, they immediately tell their collaborators, people that are part of the consortium, and before there's any publication, another lab within the consortium can check and see, is this result reproducible or not? And this is kind of a big thing because most researchers wait, they sit on their findings until they publish their paper. So you get immediate validation, and then in the next phase, what they're trying to do is see if any of the findings replicate in a new batch of cells. And so, here are some very, very basic findings. They're looking at the differences in gene expression and between the HD cells and the control cells, and what these gene differences will try to, are informative in possible pathways that are altered in HD and non-HD cells. So this kind of may highlight avenues for therapeutic intervention, things that we might not know yet, and we can do this kind of in a sequential developmental process because we're starting with stem cells, we can then generate neural stem cells, and ideally we can then generate the types of neurons that are most affected by in HD. And then very quickly they also found some other differences in the HD cells versus the controls, and all of the cells were able to generate neurons. If you stress the cells, all of the cells kind of, they don't like the stress, they all eventually die, but the HD cells were a little bit more vulnerable. And what this graph is trying to show is in the red, the HD cell is actually they're dying faster than the controls, and if you and in panel E, this is a different lab kind of running a slightly different test with the same endpoint using the same stressor. They kind of show the same thing, showing that the HD cells are also vulnerable in their lab with their readout. So this is a kind of cross-validation. And so in 2012, the consortium grant was renewed, and this format for the funding is a little bit different this time around. I know that HDSA and the HDF are contributors to this effort. This is really fantastic. CHDI also has been involved in the funding of the renewal. And here are some of the names you might be familiar with. Dr. Leslie Thompson at UCI, Drs. Jim Gosella, Marcy McDonald, Chris Ross, Elena Cataneo. They're all part of this. They were part of the HDSA Coalition for the Cure. And then there are some other people that are HD researchers, Dr. Nick Allen and Paul Kemp, Joseph Canals, Clay Svensson, Steve Finkbeiner. They're all part of this coalition, and they're all working in a very integrated manner to kind of learn more about the disease. So just for the last few minutes, one of the big challenges with stem cell research has been that we need to ideally we'd like to differentiate the cells into the relevant neurons that we think are the most susceptible in disease. So the striatal neurons are called medium spiny neurons. And the state of the art until a while ago was that you could only generate about 5% of the cells after about 100 days in culture. So it takes 100 days for these cells to mature, and only 5% of them are going to be the type of cells that you want. So obviously, any technique that actually accelerates the yield and decreases the time is something that we're very interested in. This is kind of some of the activities that the consortium is doing now. And just to highlight, the Nobel Prize was given to Yamanaka for this technique. The conversion of the fibroblasts or skin cells into stem cells using these factors was really revolutionary. And what people are now finding out is that you can actually go from fibroblasts directly to neurons using different set of factors. So this is very exciting. This is a way of really where you could accelerate the differentiation process. And just to kind of to highlight, this is a paper that was just published in January of this year by Dr. Cataneo's group, where they've already improved on the previous method for making these medium spiny neurons, which used to be over 100 days. Now these are just about half the time, and they can generate these cells, and they've really gone through a great length to characterize them and show that these cells are medium spiny neuron-like. They definitely look like the real thing, and when they're transplanted, they actually, in a rat brain, they actually take hold, and they show some of the features of medium spiny neurons. So this is very promising. We're hoping that we'll be able to generate better protocols faster. And so I'm just going to finish up here and kind of go over some of the discussion points. I hope that I was able to explain, what the difference are, that there are different types of stem cells, different sources of them, and kind of highlight, how exciting the whole iPSC discovery has been to this field. Also, as far as potential therapeutic applications, I think that the animal studies that are being done by a lot of really great investigators are really helping us understand the what, when, and where. And these type of studies are really going to help the clinicians decide what the best avenue is going to be. And then I'm also very excited about the ability of using these iPSCs in research and development so we can have a better understanding of the disease. So sorry I went a little bit over time, but I'm be happy to take questions now.
All right, thank you, Jamshid. Yeah, we have about 10 minutes, so we'll just pull up any of the questions that the audience may have. Just give us 1 minute.
Okay. All right.
So there's a question. Have there been any actual trials conducted in HD in any capacity?
So
French.
So using the stem cells, I don't believe there have been any, but the fetal transplant studies have been ongoing. There have been a number of these very small-scale fetal transplantation studies, including the larger scope, the MIG-HD, that was carried on. It's being done in France. And I'm sure that as soon as they have the results, there's going to be a big publication. We're all very interested to see what the effects were.
I have a quick question in regards to the recent Elena Cataneo finding. You said that she was able to decrease the time required to get the medium spiny neurons in half from state-of-the-art. Has she increased the efficiency in terms of raising the bar from 5% to, say, 10%?
Yes, they did. It's I think now they're getting about 10% of the cells are medium spiny neuron-like. So this is actually a large improvement. It's double what it used to be, but it's still not enough if we're to do screens or if we want to generate cells for any sort of drug screening paradigm. You need really millions of them. So we still need to decrease the time, increase the efficiency. And what this group is doing in collaboration with others within the consortium is they are developing a possible transcription factor, these factors that will help reprogram the cells from stem cell-like or maybe from even the neural stem cell-like into the appropriate neuronal subtypes. There are also, let me add on another point, there are also some interesting things being done. They are also making the accessory cells. So there's an interest from the group in making these accessory cells like the astrocytes and then you can do these elegant experiments where you combine in a dish the neurons, the astrocytes and the neurons in the dish and see if you can have an effect in the astrocytes that then translates into the neurons. These are all very exciting things that are down in the pipeline.
Okay, let's move on. That one. How good are the human embryonic stem cell studies in answering basic mechanisms of HD pathology like mitochondrial dysfunction?
That's actually a really good question. The consortium is during the renewal are actually looking at a lot of different mechanisms, and so initially in their first paper they were mostly looking at stress-induced effects. But now that we know that we can elicit those in HD versus control, the group is going to be looking at more subtle effects and mitochondrial dysfunction, energetics. There is some work that's being done along those lines.
There's a question here about HD stem cell lines. Are they federally NIH approved now?
Yeah, so the lines from Genea, the 3 out of the 4 lines have gone through the entire approval process at NIH, and researchers in the U.S. Can import them, and they can use NIH funding to work with them.
Are there any restrictions on the Genea cells for companies such as pharma companies or even CHDI from importing and using for their own research?
I think so. So Genea is making the cells available to anyone, so there's no restrictions there. And also the exciting thing is the NINDS repository at Coriell is making sure that the iPS cells are available to everyone, including pharma groups. They're trying to solicit interest from a lot of pharmaceutical companies, and just, I think, making the reagents available is really helpful to get their interest.
Since all of the cells in the human body of a sick person or the Huntington's disease carry the CAG mutation, would there be a best source for stem cells?
So no, I think that this is kind of with the iPS technology, any cells can be reprogrammed. So skin biopsies are currently done, and there are a lot of families have actually participated and donated skin biopsies for reprogramming. There are companies now that actually do routine blood draws, and they take blood cells to reprogram. So there's blood is a lot easier to get, and so that threshold, you can use those to make HD relevant cells. This is very exciting.
Is it safe to say that pre-genetic testing and in vitro implantation is a 100% effective procedure to eliminate the HD in a child?
So nothing is 100%, but this is very close to it, and it's a very effective and I believe very expensive way of eliminating the carryover of the mutation in an unborn child. So yes, I think that this technique is very, very promising. But unfortunately, I don't believe that in the U.S. the method is covered by insurance companies.
Mm-hmm. This looks like a researcher question. Can investigators outside of the consortium apply for grants in the case of CHDI contracts for research?
Yes, of course. We have a portal at website, chdrfoundation.org, and my email is available down below on the screen. If you'd like to email me about the research proposal, we can.
Great. So there's a number of questions. I'm just going to kind of paraphrase it to maybe in closing, since it looks like it's 1 o'clock. Care to speculate, given the state of stem cell and iPSC research today, when, I know it's difficult, but people want to know when you think a disease-modifying trial may potentially commence? Wow. Is that impossible to answer at this point?
It is for me.
Yeah.
So I'm not going to hazard a guess and put a timeline on it. What I'd like to really point out is discoveries are really, really unpredictable. Just like the paper by Shinya Yamanaka on the generation of iPS cells were so seminal and really revolutionized the stem cell field. So I'm looking forward to seeing those kind of research papers come out in the HD field, kind of tell us, what kind of mechanisms to target, and especially the what, when, and where. If we can narrow any of those down, I think we're going to make some really great progress.
That's great. I do have one last question that came up when you're discussing the iPSC consortium. I know there are several stem cell consortiums out there. Parkinson's disease, ALS also have a stem cell consortium.
Right.
Is the Huntington's consortium in regular communication with these researchers to try to learn what their successes or failures have been, to kind of share information and learn from each other?
Absolutely. So actually, some of the consortium members are participate in the other consortia and do some collaborations in the other disease fields as well. In addition, NINDS every year tries to have a meeting where they bring together all 3 consortia together to kind of update each other and all the other groups with the progress that they've made. So in the past, these meetings have been in DC, and I believe that they're trying to schedule one for the middle of this year again, and I'm looking forward to attending and actually hearing what progress the other groups have made.
Great. Well, thank you, Dr. Arjaman. I really appreciate you coming to our offices and presenting on this really timely topic. That concludes our webinar for today. I just want to remind everyone that the slides and audio will be available within a week on our website, and you'll soon be getting an email reminder about upcoming webinars. Next month, we're happy to announce that Holly Kardashewitz from ISIS Pharmaceuticals will be providing an update to the community on the status of the antisense oligonucleotides, or ASOs, and their preclinical testing in HD models.
Thank you very much.